Hello, welcome everyone to the first Tarim Seminar. My name is Zeynep Aydin and I am a student of molecular biology, majoring in developmental and stem cell biology at the University of Heidelberg and currently doing an internship at the Whitehead Institute for Biomedical Research in affiliation with the MIT. It's my honor and pleasure to be the first host of the very first Tarim Seminar and to cover the topic which inspires me the most, which are the stem cells. As you know, today's topic and today's theme is the stem cells and their potential, and I hope we will also discuss about how far we are from growing brains in a dish, so to grow brains artificially. For this, I'm going to cover certain topics and questions, which are, for example, the progress and potential in organoid research, organogenesis in a dish, or the generation of cerebral organoids from human pluripotent stem cells, or the modeling and development of disease using organoid techniques. So the outline is actually that we are going to start with stem cells first and then understand and explain what makes stem cells up, what their characteristics are, and then we are going to talk about organoids in general and see how they're generated and how they can be used and what their function is. And finally, we are going towards combining these knowledges that we will get from the first part of the seminar, and we are going to explore what cerebral organoids or so-called brain organoids are. So to start with stem cells. So stem cells, I can sum this actually up very short and briefly by saying that stem cells have the potential, that these are the cells who have the potential to generate every other different cell type in the body. Therefore, stem cells are cells that can generate certain cells with different functions, meaning, for example, intestinal cells, liver cells, cardiac cells, nerve cells, blood cells, or even muscle cells. And our body needs to have those different cells with different cell types in order to function properly and in order to conduct certain functions and physiological functions actually. And all these different cell types are originating from stem cells. And you can also see this by looking to this comic or this meme where we can think of stem cells to be the cells that can be anything they want when they grow up. But to characterize stem cells a little more, as we already covered, so stem cells are undifferentiated cells, but they have different and more characteristics as well we need to remember. So they have the capacity to do unlimited cell division, or at least some of them have, and while some in a little more different stage of the stem cell niche or stem cell differentiation, they have limitations, but this would go rather into detail, which I will not cover. But stem cells have the ability to undergo unlimited cell division, and they can proliferate wildly, which is actually very necessary in order to keep a stem cell pool. But if we imagine that we have only cells who conduct cell division and then proliferate, we would end up by having cells which are all the same. So we would only have stem cells. However, as we already explored, we need to have cells that conduct terminal, functions. And in order to do so, the stem cell system has developed something very smart, which is the asymmetric cell division. So we have one mother cell, and this one mother cell divides into two other cells. Well, one of those cells is kept for self-renewal, so to keeping the stem cell pool alive. The other cell can undergo differentiation, and this is necessary to conduct terminally, differentiated, to generate terminal differentiated cell types who then conduct different terminal special functions. So the next question we would ask ourselves is, okay, these stem cells seem to be very nice, have great potential. So they're the origin of all different cell types ever found in the body. But where do we find them, or where can we find them, and how do scientists actually work with those cells in labs? So there are two different approaches to generate stem cells, which the first of them is are the embryonic stem cells. And the embryonic stem cells, it's kind of the more controversial approach, because we're making use of a fertilized egg, or the zygote, and after this zygote reaches a certain stage of also cell division, the inner cell mass of the blastocyst, which we call this cell cluster at this stage, the inner cell mass of the blastocyst is taken out and plated into a dish with fibroblast feeder cells to give them, the necessary nutrition and support to keep them as stem cells. And then we would actually have our embryonic stem cells, which we can then differentiate. So once we have the inner cell mass of the blastocyst, these are cultured as said in the dish. So they're cultured, and by adding or giving them certain signals or signal molecules, we can force them to differentiate. Well, actually not force, but we can give them a direction to what they should actually become. And this can be adipocytes, neurons, macrophages, or smooth muscle cells, or astrocytes and oligodendrocytes, and a different combination of different growth factors and proteins and signals and peptides. They all, generate different cell types. And there are certain protocols for each different cell type that we can generate in labs. So the other way to get stem cells is the method with the induced pluripotent stem cells, for which also a Nobel Prize was rewarded. And this works in a way that we have a fibroblast taken from a patient. So fibroblast, an already differentiated cell which conducts a certain different function that is actually terminally differentiated. And we can take this cell from a patient or from anyone, and then give or introduce certain factors who then activate and reprogram the differentiated cell into a pluripotent stem cell stage. And once we reach this pluripotent stem cell stage, the stage where the cell has again this stem cell potential, we can go back and again differentiate out of this one cell type several different cells. This actually gives us the advantage that we can do this for every human. It's not ethically controversial, or it does not have as many ethical concerns as the method with the embryonic stem cells. And it works also very well. It has some restrictions, but the scientific community is further and further working and focusing more on this approach to enhance and elevate this current available method. Well, so now that we understood what stem cells are and their potential is, we can go further to the next topic, which are the organoids. So organoids organoids are miniaturized and simplified versions of an organ produced in vitro. So in vitro meaning produced in lab or in unnatural conditions. So these are produced in vitro in 3 dimensions that show realistic microanatomy. And these structures, as I said, they resemble the whole organs, and we're able to, or different approaches started to generate, for example, kidney, brain, liver, heart, and eye organoids. And these are actually very cool and very interesting 3-dimensional tissue culture systems. So scientists used to work with 2-dimensional culture systems until now, which is about to change, or which already changed since 2010 by introducing those organoids and by adding a third dimension, which actually makes the whole science or the whole work that scientists do on cell culture systems more translatable to the real human state without intervening into the human. But I will talk about that later. And so these organoids, these three-dimensional organ-resembling models, they are derived from pluripotent stem cells or from organ progenitors, and these can, as I said, self-organize in a three-dimensional culture, owing their self-renewal and differentiation capacities. So why do scientists actually use organoids? What are the main arguments to work with the three-dimensional organoid culture system? As I said, scientists used to or tend to work with two-dimensional culture systems, which you can see on the left, and the alternative to this are animal models. And animal models are actually very expensive. It's actually not only a matter of cost, because the mouse, for example, is used because it resembles the human as a mammalian more than other more cheaper animal models, but then it has the disadvantage of being very expensive to hold, and it also takes a lot, up to years, 2 years, until we have generated a certain mice that we want to analyze on certain characteristics and genes. And so organoids actually give us the ability, or give us the advantage to do accurate studies, since they are very close, or they're resembling the organs, but they're non-invasive. So that means that we don't have to harm or any animals or humans, and we can actually have very translational results as well. And this also comes along with the idea that they are more accurate of human development and disease than to certain animal models, because at the end of the day, animals are animals and humans are humans, and we would generate those organoids by using human tissue samples most of the time, which would translate much better to the actual state of the model organism, in this case the human that we're interested in. Furthermore, they are also amenable to live imaging techniques, which also gives us a great advantage, because if we think of three-dimensional models or in vivo models like the animal models, we can't actually image those properly, whereas I'm going to show you later how great organoids are used for this purpose. Again, so by using organoids, we can actually study, especially when talking about development, we can actually study stem cell differentiation, cellular movement, and cell-cell interaction. And these fundamental processes are very important to understand, and unfortunately are not well understood until now for certain tissues. And we can, by, for example, having greater resolutions for microscopy, or since they are more amenable for imaging, we can understand them much better and manipulate them also much more easier, and study what their functions are or how they function. We also use organoids, or organoids have a great use, as I said, for tissue biology studies. We can use them to understand which signals are secreted and which signals are taken up by which cells on a much higher resolution. But except for doing fundamental research, we do also study disease mechanisms. For example, well, let's start at the beginning. So when we try to model a disease, we, let's say, have a cohort of patients, and we take their stem cells by they're like making their induced pluripotent stem cells, as described earlier, and we then can analyze, for example, how this disease works, to do functional analysis, and by having greater cohorts, we can generate greater organoid biobanks and do wide drug screenings, which are also great because the trial to do drug screenings are very long and exhausting and also very risky, and there are different arguments that animal drug screenings are not accurate enough, and this would also overcome this whole problem of animal experiments at all. And then we have these personalized medicine, which is a very new and approach that more and more scientists and the whole medical industry is going towards. That is that we are actually able to test if a certain drug that it was developed, or if a certain combination of drugs do have a higher efficiency in this patient's organoids that we generated from that patient before testing on the patient. That's actually the idea behind it, and this is where the whole industry is going towards, making medicine much more efficient and individualized. But it's also important for regenerative medicine. So we can also, for example, take cells from a healthy donor and do transplantation assays, or we can take the cells from a diseased patient and use this unique technique called CRISPR-Cas, which I guess also most of you might have heard about by now. And we would then be able to correct this mistake in the genome of this patient, and then grow its corrected respective organ and transplant it into the patient that it was derived from. So these were some of the approaches how organoids can be used in future. And then I want to show you some pictures, because I talked a lot, but I also, like the first picture I'm going to show you on the left, it's not a picture that I have taken, it's from the internet, but the second one is animated and I'm going to play it. So you can see this is an organoid, an image of an organoid that I generated in lab. It's not that difficult, it's not that hard, it's a nice protocol that works because it's not complicated. It's these were intestinal stem cells or organoids. As you can see, this is how they look. And if you wonder how we do them, so I did organoids from isolated progenitors from the mouse. And if you don't want to see, like, you can turn off your screens because there's a graphic content ahead. Anyway, so we kind of like opened the mouse and took out the small intestine, washed it, and followed a certain protocol where we just isolated the progenitors. After that, once plating them and growing them, we worked further with our stem cell, or with our organoid culture actually. So again, organoids are used particularly for disease modeling, which we then can, for example, test drug efficacy, also drug safety testings, and the future, and hopefully the future is going towards the direction that we can actually create patient-specific organs, which can then replace organ transplantation from other patients, and patients can actually replace their own organs by generating patient-specific organoids. Hopefully then not organoids but real organs. So yeah, after you guys also know what organoids are, have a broader understanding of what is actually all possible, what organoids are used for, I want to draw your attention to one particular organoid type, which are the cerebral organoids. And cerebral organoids, I have shown two pictures. The first on the left is how they look, just very easy with the eye. They're only a few centimeters big, and the second one is higher, like a very big magnification of one of those organoids, where we can see the inside stained with different markers. So purple, there are the stem cells, and green, they're differentiated other neuronal cell types. So as you see, also in organoids, we have a big stem cell pool that keeps growing or maintaining those structures. What you can see here is it's actually very beautiful because I found this and I wanted to share it because even when we take, for example, an organoid, or if we generate organoids from a normal person, these organoids have a normal size, 4 organoids respectively. But if we, for example, generate brain organoids from a diseased patient with microcephaly, it is obvious if you compare those two brain organoids from these different origins, you can obviously see that this derived from the microcephaly patient has, like the organoid has a smaller, even the organoid has a smaller size and is brightly different structure as well. So you can see how beautifully these organoids resemble the actual state of each patient individually. And how do we generate brain organoids? Well, it's actually following a protocol that starts by day 0 with human pluripotent stem cells, growing them, then culturing them in so-called embryonic bodies. And after that, these cells are, or these embryonic bodies, which are kind of an intermediate between stem cells like human pluripotent stem cells and more differentiated three-dimensional structures, they are transferred to a different culture dish and then embedded into matrigel, which gives them a different environment where they can grow further into 3 dimension, and then they're transferred to this spinning bioreactor, which gives them the resembling of the blood circulation, and then they just grow for themselves. So it's actually not that complicated. It has great impact. To be also realistic, there is a certain problem with cerebral organoid organization. So if we look at the image on the left, we see a Lego plane that has each of the compartments of a plane, like the wings, on a position. But let's say this picture on the left resembles the real brain, the in vivo brain, and an organoid might actually look like more what is shown on the left, on the right. So we do have different compartments, we do have different, for example, cell types that are necessary, but they are not in the right position, and therefore this whole brain cannot function for itself. And this is also to calm a little bit all those historic people who claim that all organoids, we're generating brains in a dish. Where is this all going? Well, it's like, when are these brains are going to take over, or whatever questions or concerns they have. So the actual state of arts is that we can generate brain-like structures, to be more precise, in lab, but they do not function because they're really out of place. So they grow at different positions, and to get to the real state of the brain, there is like a wide or long way to go yet. And it's not impossible though. So the outlook would actually be that we can improve, for example, the bioreactors where we grow them, or we can simply create vascular networks to supply them with oxygen. And some groups already showed that it's possible, and they're really broadly working on that. Or we can do co-culture systems where we have different organoids and different cell types that give those organoids more signals to resemble their real in vivo state. And then what's also very interesting is that there are OrganChips, which connect different organoid types, for example, intestinal and brain organoids, and then they study the interaction between those different organoid structures. In particular, or just imagine they can generate a bio or an organ chip where every organ is resembled, and that, like, you see, it's going somewhere very interesting and controversial. And then there's also the spatial positioning using 3-dimensional printers, a spatiotemporal control of the signal, which would give a better resolution, but also a better control of which cells to activate at what time and where. And if we could have some topographically structured scaffolds to plate the cells on, that would also give us a better idea and a better understanding of how certain things work in the human body by using organoids. And yeah, that's it so far. So it's going somewhere very interesting, and all these concerns, I mean, they might be exaggerated by now, but it's definitely going somewhere, and there's a lot of research going on around this whole issue, which is very exciting. And with this, I would actually like to end my presentation, and I'm happy to take your questions, especially those concerning or about your concerns about ethics. If you have, if you have a broader interest in this, we could also do a joint seminar with one of my friends who is doing a PhD in bioethics. So yeah, let me know if you have further questions, and thank you very much for your attention. I hope I could explain the whole topic in a manner that everyone understood. And yeah, thank you very much for your attention.